Okay, dear friends, here we are again on the AtheroNET CardioSCOPE webinar session. Today webinar is titled Circular RNAs, How to Study Their Regulation and Function. And the presenter of today's webinar is going to be Fabio Martelli. Dr. Martelli is a biologist and the director of Molecular Cardiology Laboratory in San Donato Polyclinic. He graduated in biological scientist at the University of Rome La Sapienza and obtained a specialization in biotechnological applications. He spent some time in USA at Harvard Medical School in Boston. He worked as senior researcher at the Vascular Pharmacology Laboratory in Rome. From 2007, he has been director of Molecular Cardiology Laboratory of Policlinico San Donato. In 2017, he received the national qualification as full professor of molecular biology, as full professor of applied biology. Regarding his research, he is mostly focused on cardiac and peripheral ischemia and heart failure, and also on non-coding RNAs, such as microRNAs, long non-coding RNAs, and circular RNAs. So far, he is author of over 130 scientific articles in international journals, with H index of 52. With that, and without further ado, I present you Fabio Martelli, my dear collaborator and friend. Please, Fabio, enlighten us with the story of how we can study circular RNAs and what is their relevance in cardiovascular disease.
Thank you very much, Miron. And I don't know where you took all this information about me, but they were all right. And and I thank you and the other organizers for giving me this opportunity to talk about one of my favorite topics, that is circular RNAs. And I'll try to talk about the main techniques we use to study this RNA species, trying to give you examples on how we use these techniques to tackle specific questions in my lab. So many of the slides that you will see today are lifted from this review. That is a product of the AtheroNET and CardioRNA cross actions, and actually with several components as authors, that is about to be published as it has been accepted in British Journal of Pharmacology. So just a recap on non-coding RNAs. We know that only a small portion of the human genome is used to codify protein coding RNAs, but the large majority of the genome is actually transcribed in non-coding RNAs that can conveniently be classified as longer or shorter than 200 nucleotides, and these are called long non-coding RNAs and small RNAs. Among the small, the most studied are microRNAs, and among the long today we will focus on circular RNAs. The circular RNAs have been known for many years, first identified in 1976 as the genome of a virioid, so a pathogen. Then they were also observed in eukaryotic cells, but for many years they were regarded as either oddities or byproducts, mistakes of the backsplicing system. So only recently, considering the large amount of transcriptomes that were over and over transcribed, it started to become clear that thousands of different circRNAs are transcribed, that some of them are even more abundant than the linear counterpart, and that they are also regulated independently from the linear counterpart. Then people started looking for function and they started to find more and more functions associated with the circular RNAs. More recently, it has also been found that some of these circularRNAs are actually translated in mini proteins. Once again, it's a story of junk that is not so much junk after all. So biogenesis, there's more than one model present, and these are the three main models. So how they are generated. So the circular RNAs are the product of a so-called backsplicing event, so that the 3' of an exon, instead of being joined to the 5' of the following exon to generate a canonical linear RNA, is actually backspliced, meaning fold back, and the 3' binds the 5' of a preceding exon. And this can be driven by RNA binding proteins binding the intronic sequencing and facilitating this backsplicing event. Also inverted repeats that can facilitate the association of intronic sequences can facilitate this event. And if there is more than one exon, whether or not the intervening intron is spliced makes the generation of a fully exonic circular RNA or an intronic exonic circular RNA. And from the lariat form during the splicing, you can have that the lariat is not efficiently debranched, and the tail is trimmed, and then a circular intronic RNA is formed. Circular RNAs containing introns are mainly nuclear, and for some of them functions of regulation, the transcription have been described, but the majority of circular RNAs are exonic in nature, are mainly present in the cytoplasm, and they mainly work binding either RNA binding proteins or microRNAs. Although, as I said before, for a small portion of them, very small portion, it has been in a convincing way demonstrated that they are translated. Just a small slide to remind just what microRNAs are, because microRNAs are an important element in this story. They are small endogenous non-coding RNAs, about 22 nucleotides in length, that act by binding the target RNA, mainly mRNAs, inducing the degradation of the target through the recruitment of a risk complex and or the repression of the target. So one feature of circular RNAs is that they are devoid of 5' and 3' ends, so they are less sensitive to exonucleolytic degradation, and this potentially make them good markers, as demonstrated by our friend Ivan Devaux, that, for instance, identified this circular RNA as downregulated in blood cells of patients with left ventricular dysfunction. So now I will go over a variety of techniques that can be useful to analyze circular RNA levels and functions. So one first point is detection using high throughput system that is very relevant for cost action that is about omics. So there are, I mean, the name of the game is always using the only element of the circular RNA that is really specific for the circular RNA and not shared with the linear counterpart. That is the back splicing junction. So one way is to use arrays with oligos that are actually complementary to this back splice junction. They are on the market, but not widely used because, of course, as all arrays, they become old very rapidly. And the other thing also, the stability of the signal is suboptimal, let's say. So the most highly used for high throughput detection is actually RNA sequencing. But once again, you cannot use all the reads that you have. You can only use the reads that actually are across the back splicing junction. And so you can figure so that you can use only a very small portion of the available reads. So there are techniques to try to increase the percentage of these reads. So number one, you cannot use a large amount of RNA-seq data sets that are in GEO, because they were generated using poly-A enriched libraries. So CircularRNAs are by definition excluded. Ribodepleted libraries are very good. You can also associate ribodepletion to also depletion of poly-A RNA. That also helps. And another technique is trying to degrade as much as possible the linear RNAs, treating with an exonuclease. This is an efficient technique, but it is also prone to artifacts because, for instance, a large circular RNA that has a nick is now seen as a linear RNA and degraded by the exonuclease. Regardless, the technique that you use, the factors influencing circular RNA detection are the following. So library preparation, as we said, is very important. Read depth is the most important parameter. So we don't have many reads, we have to go very deep. In our experience, with less than 100 million reads per sequencing, you cannot do much. And the other thing is using enrichment methodologies, as we said, and also using the longer the better with the reads length. Okay, so then crucial for the identification of circular RNAs is using the right bioinformatic tools. I'm no expert, and if you have questions, you have to ask Spiros or other bioinformaticians, but more or less these are the underlying principles. As I said, for instance, this is exon 1, exon 2. Let's say that there is a circular RNA formed. You can only use a splice, back splice reads, so those that are across the junction, like this one, while this one is actually describing the forward splicing, or a read that has some of the exon and some of the intron is describing the precursor RNA. So there are several tools that have been designed to detect circular RNAs, CircExplorer, Ciri2, and others. All of them have plus and cons, so our advice is to use more than one in order to identify sort of a core data set, circular RNA set that is more reliable. Then if you want to really quantify circular RNAs that are also softwares to do this, they are not as efficient as those that we use for differential expression with linear RNAs. So again, high depth helps a lot as much as having many replicates. Still, validation is really very important, if not mandatory here. Much more accurate, much more, many more RNAs you have to validate compared to linear because they are much less reliable than the software to identify differentially expressed linear RNAs. And these detection tools tell you about the junction, but not how the circular RNA is composed, whether there are introns or intervening exons are present or not. So this there are softwares again to try to do this. Again, they can be very helpful, but validation. And then very important is databases. Circular RNAs are not annotated in ensemble or so we have to rely on circular RNA databases such as CircBase, CircAtlas. Some of them can also help in the identification of interacting RNAs, and also some are useful to link circular RNAs to disease, including cardiovascular disease. Okay, this was about throughput, high throughput techniques. I said that validation is of paramount importance, and the main technique for validation is qPCR. So how you can detect it very easily using divergent qPCR primers that can only generate an amplicon if they are if the RNA is actually circular. Then you generally design also another couple, generally with a primer in common that is actually in the next junction on the linear RNA that by using regular convergent PCR allow you to measure also the linear species. So this is very important element, because also by measuring the abundance of the circular and the linear, we can have important elements. So we can say whether for instance, a circular RNA is modulated, whether also the ratio between circular and linear isoforms is also regulated. It tells you that the circular RNA is regulated independently of the linear. And it is not just, for instance, increasing because you have more expression of the circular of the locus, and this generally independent regulation is a telltale of a function. Then characterization, as I said, it is also important to sequence not only the back splice but also the wall composition of the circular RNA. Again, with a well-designed divergent primer couples followed by standard sequencing. And here I can show a real-life example of why this is important. We identified this circular RNA that is induced in hypoxic endothelial cells. So there are three isoforms of this circ anchor D12. One that is composed only by the exon 2 and exon 8 that are the backsplice junction exons. Others are that are also notated are composed of all the exons between the 2 and 8, and another isoform is missing exon 4. So by putting correctly the different primer couples, you can identify all these species and find which one is really expressed the most in your system. For instance, in endothelial cells, these two were expressed more or less at the same level, while this one was essentially negligible. Then you can also have surprises. For instance, this is a CircBPTF, that is a CircularRNA expressed in the failing hearts, that is composed by exon from 21 to 27. And this is the backsplice junction. But we found that in the heart, there is an alternative version of exon 21 that is essentially missing half of the exon. So this is very important. Also because, as we will see, CircularRNAs most often work by binding microRNAs or RNA binding proteins, and if a sequence is or is not present, of course, makes a big difference. So in the characterization, it's also important to really reassess the circular nature of the RNA, and you can do that by taking advantage of exonucleases, for instance, RNase R, that, as we said before, can cut the linear but not the circular isoform. And this again is how we characterize again CardioSCOPE. So the linear isoform is resistant, while the linear isoform was degraded upon RNase R treatment. Then there are other systems to detect. There are other systems to detect circular RNAs that that do not take advantage of a reverse transcriptase. So these are sometimes advised when the quality of the RNA is quite low. So of course there is the good old northern blot, and by placing strategically your probes, you can target again the backsplice junction, an exon composing the circular RNA, but it is also present in the linear isoform that will show two bands, or an exon in the linear that is not part of the circularization process. And then there are other techniques that I have to say are not very frequently used, such as nanostring, where you have a capture a probe that is biotinylated and a probe that, for instance, recognizes the junction with a unique fluorophore-based barcode, and using the same capture probe and two reporter probes, one for the junction, one for an exon that is not present in the circular RNA. You can detect using the same capture the two, the linear and the circular. And splint-seq, you have two essentially sequences, two probes that bind to the two parts of the backsplice. Then you have a ligation, and then you can have adapters that can that can be amplified only if somehow the circular RNA acted as a scaffold. But as I said, these two techniques are seldom used. And then you also want to see your circular RNAs, and you can take advantage of in situ hybridization. I have to say, it's not they're not easy. They're not easy techniques. And one is just to design a probe on the backsplicing, conjugate it to a fluorophore. Then you can use the base scope technique that in order to try to amplify the signal a little. These techniques are not very sensitive, and also you don't have much choice to locate your to put your probe, because you have to target the backsplice junction. This works with fixed cells. In vivo, you can use a system with a GFP tagged and does detectable by fluorescence version of Cas9 or Cas13, and of course that is catalytically dead, and then you can have and then you need an sgRNA that again targets the backsplice junction. This is suitable as a not easy system to set up, but it can be used in vivo. And then there are more, let's say, futuristic approaches, very not really in use yet, such as the use of molecular beacons and amplification systems using a streptavidin binding system of fluorophore label oligonucleotides with them trip technique. Then another thing I said for exonic RNAs is that they are mainly located in the cytosolic fraction. So this is another important control you have to do. So you fractionate your cells, and then you see where it accumulates. For instance, these two that are CircPVT1 and CircVPTF that are again expressed in failing hearts, they accumulate mostly in the cytoplasmic fraction. In this case, nothing particularly sophisticated, but again, very important to have proper controls with it. So to study the function of circular RNAs, one of the most widely used technique is actually the good old silencing using small interfering RNAs. In this case, again, you have to be able to identify a siRNA that targets the junction, but of course does not target the linear. And this is, for instance, the example again of circ ANCAR-D12. So you have to generally screen a few before you find one that really works in inhibiting your circular RNA isoform, but it is not effective on the linear. You also have to be careful that the 5' of the siRNA does not bind to your linear and works in this way as a microRNA inhibiting translation. But generally you have to work a little, but generally works. Another way is just to use the Cas13 as typical of this system using guide RNAs and Cas13. And again, the gRNA has to target the junction. Knockout techniques can also be used. So you can, for instance, remove the intron so that the circular RNA, so that the end is no longer available for backsplicing. Or you can remove the sequences that, for instance, the repeating sequences, the general ALU sequences, or the RNA binding protein binding site that drive the circularization. However, introns are tricky elements to touch. There are a lot of regulatory elements that are in, such as, for instance, enhancers that are in introns. So this is not always possible. Then there is the gain of function approach. This is so overexpression. One way is just to express the sequence that needs to be circularized, along with inverted repeat sequences that facilitate the formation of the circular RNA. This works very nicely on paper. However, in real life, this process is present, but it's not very efficient. So it's very important to have as controls expression vectors that are exactly like this, but without one or both the circularization cassette, because it's a circularization efficiency is generally is not always high, let's say. For shorter circular RNAs, you can also generate those in vitro by in vitro transcription, followed by ligation by T4 RNA ligase. Here, it is very important to have a good purification protocol, because the ligation process is efficient, but it's not 100%. So you can have and we know that the linear RNA is much more efficient than the circular counterpart in the activation of the innate immune system that trigger interferon response and cytokine hyperproduction. Regardless of all these difficulties, we can definitely envision the use of loss or gain of function approaches for cardiovascular diseases, using the typical tools to deliver them. So viral vectors, mainly for expression vector for circular RNAs or for shRNAs, nanoparticles, exosomes for CRNAs, for instance. And then it starts, the things that we have to say are valid for all the protocols of RNA therapy. So the issues are efficiency of delivery and the specificity of delivery. So to date, we are at the stage of the preclinical stage, but hopefully we will soon move to humans. And this is again a slide lifted from the review that summarizes these processes. So we have first identified the circular RNAs that are deregulated in the state your study. Here is for the heart, but it can be atherosclerosis or other diseases. Important is also modifications of the RNA. For instance, MSCA modification plays a very important role in circular RNA physiology. For instance, it has been observed that if you synthesize your in vitro your circular RNA, having MSCA modification makes the RNA much less prone to trigger a response from, as we said before, an immune system response by the innate system. And then you have to go over again with the identification of proper tools to modulate your RNA. Then there is a lot of design, optimization, synthesis, delivery issues to be solved, and then you can finally come to preclinical first. And as I said, we are at this stage, but then finally. Often we will hopefully will be able to bring this to clinical practice. These are just some examples of the circular RNAs that are modulated in atherosclerosis. So the field is very active. The prototype of all this is Unreal, that is the product of chromosome 9 p21, that is very important for many diseases, but also for atherosclerosis. And the balance between the linear and the circular isoform is very important for atherosclerosis. The linear isoforms of Henry are proliferative and anti-apoptotic and pro-adhesion for smooth muscle cells and facilitate the formation of atherosclerosis, while CircularRNAs has the opposite effect, and it works through the binding of an RNA binding protein, pescadillo, that in turn is important for RNA, ribosomal RNA maturation, so affecting nuclear stress and the activation of P53 pathway. And now I would like to provide you a few examples of how we took advantage of all these techniques to just study a few circular RNAs. So here is one of the topic we really investigate in our lab. So what comes after atherosclerosis in the coronaries goes bad, you have myocardial infarction, and if the patient survives, then remodeling starts, and that is characterized by extensive remodeling that is structural but also molecular. And if things again go in the wrong direction, you start the patient has heart failure that can have very severe or even consequences or even death. And so we said that we wanted to study this moment in heart failure where all the decisions are taken. And to do that, we took advantage of the possibility to take the remote zone of the ventricle in ischemic heart failure patients undergoing ventricular restoration procedures in our institute. So the aim was here to identify networks that are constituted by circular RNAs, microRNAs, and mRNAs, constituting a so-called competing endogenous RNA network in ischemic heart failure. And how it works like this. There are circular RNAs that can bind microRNAs, and this way the microRNAs are inhibited in their action of binding and inhibiting their mRNA targets that are in this manner derepressed. So to study this RNA networks in heart failure, first we started generating data sets by RNA-seq of these left ventricle biopsies of failing or non-failing patients. We generated an mRNA data set, a circular RNA data set that was extensively validated, looking for those circular RNAs that were deregulated also independently of the linear isoform. And then we also looked for microRNAs that were deregulated. And then we applied this analytical pipeline. Maybe we don't have to go through all the passages, but essentially for every predicted interaction, we have, let's say, a reality check, meaning the check of these data sets that we generated that really say what are the circular RNAs, the microRNAs, the mRNAs that are deregulated in heart failure. And this way we identify this CERNA network that looks like this. So at the center there are circular RNAs, then there are the interacting microRNAs, and the mRNAs regulated by one or more than one of these microRNAs. And the gene ontology analysis of this deregulated mRNA tells that indeed this RNA CERNA network regulates important pathways in heart failure pathogenesis, such as we identify categories related to prototoxicity, as well as mTOR, FOXO signaling, and MAP kinases. And then we wanted to look in further detail how to really take advantage of this information generated by this analysis. And we, for instance, here there is the case of one of them, circular PBT1. It is very simple structure composed only by one exon. We targeted by CIRNA using all the care that I told you about, and looked for by RNA-seq, though the differentially expressed genes, and we identified a series of interesting categories, but really what got our attention was TGF-beta regulation of extracellular matrix, because this could mean that this was involved in fibrosis, that is very important pathogenetic process in failing hearts. One element that told me that we are on the right direction is that indeed cardiac fibroblasts treated with TGF-beta, we found that circular PBT1 was induced while the linear counterpart was not. And then we looked what happened to the TGF-beta response when CircularPBT1 was silenced, and we found that profibrotic markers such as alpha-SMA and CTGF, their expression upon TGF-beta treatment was attenuated, and also the pathway of TGF-beta was also attenuated. And then we said, okay, we have to identify how it works. So the network identified one important microRNA 30, but we wanted to see whether this analysis somehow one other microRNAs escaped this analysis. So what we did was doing a pull down using a biotinylated oligonucleotide targeting the splicing junction, and then sequencing what was pulled down. In this way, we identified two other microRNAs that are involved in potentially in fibrosis. After extensive validation, we looked at the function. So what we expected is that among the target genes of this, microRNA should be enriched among the downregulated of genes when we silence CircularPBT1. And that's exactly what we found, indicating an antifibrotic function of these microRNAs. And these are somehow a busy slide, but this is a rescue experiment. Essentially, let's look, for instance, this one. We have that CTGF is induced upon TGF-beta treatment, less so if we silence CircularPBT1. But if we also inhibit the microRNA 125, we have a rescue of a full capacity of inducing CTGF by TGF-beta. So this proves that 125 and microRNA 30 are indeed capable of rescuing the silencing of CircularPBT1. So to summarize, we identified a CircularRNA signature characterizing ischemic heart failure, and a network composed of CircularRNA, microRNAs, and mRNAs that is deregulated in heart failure. Okay, we can skip this, and this is CircularPBT1 is in particular induced in TGF-beta, by TGF-beta is an element of a profibrotic response triggered by TGF-beta, at least in part through the binding of microRNA 125 and microRNA 30. This is the most important slides. Most of the experiments on circular RNAs were done by Alessia Bibi, Alicia Madin, and in collaboration with Simona Greco. And I also thank Spiros for his expertise on bioinformatics. And thank you for your attention. Shall I stop sharing?
Yes, I mean, you can. It's going to be easy for you to follow the discussion and to watch us. Thank you so much for these insights in the CircularRNA world. It's very exciting. It's very novel, I would say. And thank you for really summarizing all the details, the technical details, which is important for new people that want to really engage in these types of studies, but also showing some of the results that really show the relevance of CircularRNAs. We can start the discussion. Let me be a good host and then ask others if they have questions, because I have tons of written down, but I will let others join the discussion if there are any questions or no.
I have a question. Fabio, I have a question regarding the last part of your presentation, the CPVT1 story. So you said that TGF-beta treatment induces CPVT1, but CPVT1 is an inhibitor of TGF-beta, right? Am I correct? So this is a
it's actually no, it's profibrotic. So what I mean, I understand it's a little bit a convoluted way, because what all the experiments I showed you are loss of function experiments, meaning if you inhibit the circular RNA, you have that the response to TGF-beta is less strong, let's say. And it does that, at least in part, by binding these microRNAs that end up being antifibrotic. So when you remove the circular RNA, the microRNAs are more free to do their job and somehow attenuate the profibrotic response. And in this respect, at least in theory, this makes also an interesting therapeutic target for heart fibrosis.
So you believe that there is only one mechanism for its circular RNAs? So in this case, there is only the sponge function. There is no other function?
No, I don't think so. That's the one that we explored because we are an RNA lab, so it was easy for us to explore. But of course, they can also bind RNA binding proteins. And this is another thing that it's on the to-do list because we know that they bind RNA binding proteins, that they are important, and probably also in conjunction with microRNAs.
And one final question, if I can, what is the concentration of these circular RNAs in the cell? I mean, relative to other RNAs that are present there?
Yeah, that's not easy question that you're asking, and I can say that it is readily detectable. That is, so its expression levels is comparable to that of many other mRNAs. Is a bit more expressed than the linear counterpart. So it's more abundant the circular than the linear. But if you ask me if we had made absolute quantification, we have not. So I cannot really tell you. but it's a mid-range, let's say, expressed RNA, looking at the number of reads and looking at the number of the CT values using primers with certified efficiency.
Right. Okay, thank you.
We have a question in the chat from Noemi, which kind of lines up with what Dimitris is asked. So she says it might be a basic question, but I don't know it. Which is the percentage of CircularRNAs in the circulation in comparations to microRNAs? How abundant are they? So now we move to the circulation, which I think it's even drastically more challenging.
Yeah, it is difficult. I mean, the stoichiometry is something always somehow difficult to really address, and very few papers do that. Let's say if we go in a very more, let's say, down to earth and very practical thing, how easy it is to detect them. I would say that top is microRNAs, then there are CircularRNAs, and then there are linear RNAs, more or less. If we are talking about plasma, or they are definitely in an extracellular vesicles. Then, of course, if you look for total RNA, total blood, or PBNC, what you're detecting is cellular levels of microRNAs and the CircularRNAs as well.
Have you tried so far to measure it in a plasma or serum, just like out of curiosity to see if it's detectable?
Yeah, some of them, for instance, those that caught our interest in the heart, unfortunately were not detectable in plasma. Others are definitely, so I don't want to discourage anybody, but let's say microRNAs are way easier. I don't know how much is a technological issue and how much is the real abundance issue, because as I said, nobody really, it's very difficult to do, to have absolute quantifications.
Yeah, maybe with the digital droplet PCR, I know we previously, a couple of years ago, did something with microRNAs. It was just a pilot project that unfortunately we never went to pursue because we couldn't afford ddPCR. But for example, for microRNA, we were interested, at least my colleague was interested in analyzing microRNAs that are coming from placenta of pregnant women. And they are not really measurable by qPCR because CT value was around 36, 37. But with ddPCR, it was quite easy to detect them. So that might be some tool to pursue in the future if you have the opportunity at least to try it.
Yeah, absolutely. We have not done that, I have to admit, but it's something that once you put your hands on something promising, you definitely want to do.
Okay. I was interested in several things. First, when you discuss about this type of targets that are really low abundant, like you need to go very deep with sequencing, 100 million and more to find them. I was always curious about biological relevance, because if you look the data and then you understand that in the let's say bulk RNA-seq data, there are many cells, and then you go so deep, then basically probably if you move to single cell.
No, I understand what you're saying, but in this case you have to consider that if you have a linear RNA, you can use all the reads that can describe this RNA. So you have for a regular, let's say, linear RNA, you can have hundreds of reads because one goes on exon 1, another one goes on exon 2, and so on. Here, you can only use the reads that are on the junction. Okay. So all the reads that describe the exons constituting the circular RNA are in common between the circular and the linear isoform. so you cannot use them. So you can have circular RNAs that are quite abundant. You have a lot of reads that are actually recognizing circular RNA, but you cannot really discriminate those reads that actually are coming from the circular and the linear. Is something similar happens with when you want to do an alternative splicing detection protocol. Then you can only use the reads that are really on the junctions. It's essentially the same problem.
Yes.
So if you want to do an alternative splicing project, you need very deep data sets.
Okay.
Because it is not sufficient to see that the gene is there. You want a definition at exon level. And if you want that kind of definition, you need that. So not necessarily those circular RNAs that maybe in your data set were described by 4, 5, 6, 10 reads, then you try to detect them by qPCR. Boom, very easy. CT is 20, is something like this. Very easy to detect.
So basically, when you say 100 million reads, basically functional reads are much less than this 1 million, let's say.
It's really a tiny portion. That's why these enrichment protocols are used, because you don't want to waste 99.9% of your reads. It's not really wasted because you use for also to detect your linear RNAs. You can do also use to do other alternative splicing of the linear isoforms to have information at the exon level. So it's not really wasted, but let's say, I mean, if you really want to focus on circular RNAs, then you want to try to enrich as much as possible.
I have just another question, which is more like related to the mechanism of action, because you show nicely that CircularRNAs most of the time either interact with RNA binding proteins or do a sponging effect. Now, then you move forward and you present some of the data, and then you showed that when you bind siRNA to the circRNA, you can see that there is RNA decay. Basically, you are degrading this RNA. So I was wondering if most of the time when we read papers, we see that circRNAs are presented as sponging of microRNAs. So what is happening in cell when microRNAs bind to these circRNAs? Does then microRNA direct this circRNA to the degradation, or is there some kind of just decreasing concentrations of microRNAs?
This is an excellent question, and it's actually not really very clear what happens, because often it is described that upon binding, the microRNA levels are reduced. Or if you overexpress the microRNA, you have actually that a circular RNA level can go down. However, this is not always the case. Sometimes you have zero regulation, and you, for instance, for some of these we found that for some of these circular RNAs, the binding has zero effect on the levels of the microRNAs, but their targets at that point are de-repressed. So you see that there is a biological action, but there is no regulation. And sometimes it's even true the opposite. So the circular RNA works as sort of a storage and stabilization tool, and actually works as a sink, let's say, but to have it again available, to make it available in other conditions. So sometimes the microRNA and the circular RNA are co-regulated. You have pretty much all. And what are the determinants that make it work in one direction or the other? Completely unknown.
Okay, so sometimes when we want to do the functional analysis, we don't have the guarantee that if we use siRNAs that we are going to downregulate CircularRNAs, or because the mechanism is more or less very related to the microRNAs, or with siRNAs we always can guarantee that
I
it's
mean,
going to be
siRNA work as siRNA, so some of them work and some other don't, and we don't really know why. But when they work, they definitely use the cleavage function of AGO2, and so is clipped, and once the circular RNA is cut, then it's also degraded. So siRNA targeting the junction do their job. What the interaction between microRNAs and circular RNA is more complicated. But it's also a different interaction. siRNA work with a complete binding, microRNAs bind only with the seed or in seedless manner, many ways.
Okay, thank you.
Question.
Yeah.
Oh, maybe there's something in the chat, probably, if you want to go ahead.
No, please go, and then we
very quickly, maybe a naive question. So it looks like the dynamics, the dynamics is first of all, thank you for this fascinating presentation, because really it opens a new world for some of us, me included. So now, probably, if I got it right, the dynamics, also a sponge, anyway, the dynamics between circular RNAs and microRNAs and binding protein and so on, under physiological and pathological condition may vary according to moment by moment and probably from cell type to cell type. So we can generalize. But when these circular RNA are around, so is there a preferential binding of microRNAs to them, or this is something that can be, let's say, variable from site to site?
Okay, what I can tell you is that, I mean, these are all excellent questions and very few answers are there. I would say that for non-coding RNA, it's applicable, the rule that is applicable for coding RNA. So definitely all the functions are very context dependent and as a consequence, cell type dependent. So, for instance, the stoichiometry or even just the presence of a certain microRNA can change from one cell to the other, or in one condition, or as opposed to the other. So these are, as I tried to convey as a message, these are complex networks constituted as a minimum by 3 elements. And by touching one element, you have that the whole network finds a new organization. So, and sometimes it's not so easy to predict touching one element of the network, what consequences is going to have on other elements. For instance, if I remove the binding sites for a certain microRNA, this microRNA is now free to target more efficiently other parts of the network that is also unbalanced, so it can find a new equilibrium in another shape. This is all definitely cell type dependent and physiological state dependent as well. The details are, very few cases have been really clearly identified.
Thank you very much. Which means that it's so difficult to define the experimental conditions. Let's say with cells, it's very difficult because if you have one type of...
It's as difficult as it is for any other experiment you may want to do with mRNAs. You have to, the general rules are always the same. You try to control as much as possible your conditions, and then you describe them as good as you can in your manuscript, and hopefully someone is going to be able to reproduce them. But then you don't know if your incubator that day, instead of being 37 degrees, was 36.8, and whether or not this is important for your system is very hard to say.
We're going to have maintenance of the incubator in 5 days. Anyway.
Thank you. We have another interesting question from Amy in the chat. She says, if you know that one linear RNA is quote unquote good, can you make the circRNA become linear and then become a good linear RNA? It has been done talking about linear unreal, different role depending on which one is more present. So basically, can you convert, can you use the circRNA that is good to trick the system and make it more linear and then benefit from it?
Well, I'm not sure I understood what is the purpose of doing such a thing. What I can tell you is that circular RNAs are being explored as a biotechnological tool. For instance, to do more efficient RNA vaccines. In this case, you have to combine them with IRS so that they are efficiently translated. And again, here the idea is that circular RNAs are less prone to degradation since they don't have they are not prone to exonucleolytic degradation. Yes. And as I said, no, you have to generate those artificially. Then you have size constrictions. And another thing is good to incorporate at least some MSCA modifications or do some editing to make them as natural as possible so that they are more stable and that they trigger as little as possible the interferon response. This thing of making linear, circular, linear, I don't know really, probably did not answer correctly, but
yeah, I guess is the idea if you have like an unreal, the linear form is bad in terms of atherosclerosis and circular form is good. Can you transform circular RNA into linear form and make it good? Will just making this circular form linear retain its properties of being good per se?
I see. Yeah, well, I don't think anybody really tried that. So one possibility is that the circular RNA is but the circular RNA is not just a portion of the linear, because if that was true, that would be too simple. It's clear that functions are also related to its structure. Whether this means because certain targets are bound more efficiently or they no, we say nuclear cytoplasmic, but this is we know this is very completely rough way to see things. I mean, whether this means that it really goes better, more efficiently in certain parts of the subcompartment of the cell than other, or I mean, we know that circular RNAs are definitely in the risk complex, but how this is, if the risk complex is different, whether the microRNA, RNA couple is in a circular or a linear fashion, we ignore all this. It probably is not just a matter of what are the exons that are there. It's also important the shape that it has, because this probably allows the RNA to have we like to think as strings or circles, but of course they are tangles, right? Okay. And so the structure is probably very different.
So Fabio, the linear and the circular unreal do not have the same sequence. So it's not just one is the circular version of the other. They have different sequences, right?
For instance, for generally the circular is part of the linear. For instance, you have exon 1, 2, 3, 4, 5. You can have that exon 2 and 3 can also form a circular RNA. So you can have these two exons 2 and 3 that are present either in the linear or in the circular RNA compartment.
Okay. And if I rephrase this question, biologically, is it possible that for the cell to convert a circular to linear? Is there a nuclease, for instance, endonuclease?
Yeah, generally, when you do that, for instance, using a siRNA, then the RNA is rapidly degraded because it's not polyadenylated, it's not capped, and so it's degraded right away.
But not converted to linear by endonuclease.
Well, it is, the siRNA with the risk actually has endonucleolytic activity and clips the circular RNA and convert it into linear, but then since it is not protected by the A and the cap, is at that point all the exonucleases kill it right away.
I see. Okay.
I have another question regarding the specificity of circRNA. I mean, tissue specificity. Are there more tissue specific than microRNAs, for example, and mRNAs, or is it just the matter that in this tissue they are more present and we see them by sequencing, and they are also present in other, but we don't see them because we didn't sequence too deep? Do you have any kind of information?
So every one that comes with a new RNA species, the claim is, oh, this is more tissue specific, describes disease better. This happened with microRNAs, it happened with circular RNAs. Well, I think that the reason is the one that you said, that we know less, so we think they are more specific because we detected less. When you start digging, you see that all this specificity is not really. I don't think that there is one species, RNA species, that is really more specific or more describes inherently better the disease state. I think that you have to check. No, you have just to look very hard and identify the one, and probably like all biomarkers. I mean, it is the name of all biomarkers that. I mean, they probably work in combination. If you want to really, you have to define the limits very well, and then you, in combination with other things, they can be useful.
I was more thinking about from the side of the therapy, because if you want to, target something for, I don't know, heart failure and cardiac fibrosis, and then you end up treating, I don't know, something in the brain or something else. This is why I'm asking, because field of RNA therapeutics is now really hot topic, and it really expands a lot. Everybody is trying to discover new targets, and with long non-coding RNAs, we can see that, as you mentioned in every paper, there is this sentence about the specificity, but then you go on any single cell data and find any long non-coding RNAs. I mean, not any, but not the most prominent one, but more that are, I don't know, not well known. And then you see that they are present in adipocytes, in T cells, in astrocytes, in, I don't know, myofibroblasts, whatever. It's just a matter of how many cells in the mix have those, and the concentration per cell is the difference. So this is why I'm asking for circRNA, if it's a different story, of maybe it's just a technical thing.
I think it's the same story, meaning if we ask me if if there are different transcriptomics pattern looking at different cells, different organs, different disease state, of course there are. What I don't like is the word specific, because nothing is really specific, okay? It's much more abundant in one condition, one cell type, than in other. So let's say microRNA 122 is liver specific. Yes, but then you have that pops up in many other conditions, especially in plasma, and it goes down, for instance, upon myocardial infarction, other diseases. So where is the specificity? I mean, it's definitely, it is expressed. I mean, you can find difference in the abundance of circular RNAs. Some are more expressed in the heart, other in other organs. That is for sure true. If you want to say the word specific, that means it's expressed there and not there, then of course it goes to what you were saying before, how sensitive you are. If your threshold is here, then, and they are expressed like this, you don't see this, but not because it's not there, it's just don't see it. So the name of the game is using low sensitivity techniques, and then you will have a lot of
more specific, yeah.
a lot of specificity, but I don't think that's the way to go.
Okay, that's it for me. We don't have any more questions in the chat. I don't know if Dimitris or Paolo have any other questions. If not, we can let people start preparing for 1st of May. I guess everybody has a free day for that. Maybe somebody are going to extend this on holidays. Me and Dimitris are going to prepare for Easter on Sunday.
Yeah.
All right, exactly.
Yeah.
It's your turn to have Easter now.
Yes.
Yes, it's our turn.
Okay. Thank you so much for this. For everybody who are left with us, just to know that on the next webinar series is going to be on the 22nd of May. Noemi will present very interesting topic, new approaches to inhibit microRNAs in atherosclerosis preclinical models. So we are really overwhelming the AtheroNET with transcriptomic side of the story and with RNA. But maybe we will end up with this one and then start from the fall with maybe more proteins and metabolomics. We'll see. Okay, thank you so much. Have a nice week. I wish you all the best. Thank you, Fabio, so much, and see you soon. Bye.
Thank you. Thank you. Bye bye. Bye bye.